Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the last lecture and today I will be discussing about how to design a fluorescence microscopy experiment. Okay, so by now you have learned so many different types of microscopy techniques, the different types of illumination modes available, different types of technologies, super resolution imaging and single molecule imaging, and all different sorts of technologies you have studied. Okay, so in today's class, what I will show that how you can take a decision that for addressing this particular biological question, this technique is good enough to provide me the results. Okay, so how to select the best microscopy method for your imaging needs, that you will learn today. So whenever you have to design an experiment, first you should ask all these questions and write down the answers on pen and on a paper. Okay, so let's say first what you will ask, that what is your model system, whether you want to image bacteria, yeast, cell lines, tissue, embryo, organoids or organism, etc. Okay, so how does it matter? Because this, you can see the second question, that is how thin or thick is your sample. Okay, because depending on that you have to decide that what technology is good for your imaging experiment. Okay, so for thin samples like bacteria and yeast cells, you should generally go for wide field imaging. Okay, but for the thick samples such as thick tissue samples, embryos, organoids, or even imaging entire organisms such as zebrafish or C. elegans. Okay, for those kind of thick samples, you should go for confocal or light sheet and 2-photon illumination. So confocal is kind of an intermediate, confocal microscopy can provide intermediate depth, okay, which is lower than light sheet and 2-photon, but better than wide field imaging. Okay, but for really thick samples, like if you want to image organoids or entire organism, then it's better to go for either 2-photon microscopy or the light sheet imaging. Okay, then you have to decide that which illumination mode is ideal for you. So there are several illumination modes that that we already discussed in one of the previous class, right? The epi illumination, which illuminates the entire cell. Then confocal illumination illuminates this cone kind of provides cone kind of illumination. The TUF microscopy provides illumination very close to the microscope slide or the cover slip. Okay, and then light sheet illumination passes a thin sheet of light through the sample, and 2-photon illumination only illuminates one particular spot in your sample, and high-low illumination passes a thin sheet of light through the cell, entire cell volume. Okay, so these are different illumination strategies and all of them can be used for different purposes. Okay, let's say if you want to study any membrane bound proteins, okay, or you want to do some in vitro single molecule imaging experiments. Okay, you just tethered DNA to the microscope slide, and then you want to see some fluorescence molecules or the fluorescence beads interacting with DNA and something like that. Then for those kind of experiment, you should go for the TURF illumination. Okay, but if you want to do imaging in really thick samples such as embryos or thick tissue samples, then light sheet and 2-photon illumination helps, because they only illuminate your plane of imaging, that is a focal plane, and it avoids unnecessary photo bleaching and photo damage in the planes above and below the focal planes. Okay, and high-low illumination is used for single molecule imaging and tracking in cell nucleus or interior of the cells. Okay, so because a thin sheet of light passes through the cell and it illuminates fluorescent molecules in within the cells. Okay, so these are different illumination strategies that we already discussed in one of the previous class in detail. So you have to decide that which one is best. Okay, for regular imaging of bacteria or yeast cells, you can just go for the epifluorescence microscopy, okay, which is quite convenient and conventional technology. The next question that you should ask is, what magnification do you need? Okay, because most of the microscopes are equipped with several objective lenses. 10x, 20x, 40x, 60x, 100x. Okay, so not necessarily that if you have a 100x objective lens, so for all your imaging applications, you will just use 100x because it gives you more magnification. Okay, that is not the case. So you should decide at what magnification do you need and how you can decide that. So you have to see whether you just want to count the number of cells and see their morphology, or you want to count the mitochondria within the cells and see their morphology. Okay, so if your purpose is just to see the number of cells and count whether they are positive, negative, or they are live or dead, kind of just by looking at the external morphology kind of thing, then obviously the smaller magnification is better because you can have a larger field of view. Okay, you can see multiple cells in one image. Okay, whereas if you go for the higher magnification, you may hardly see few cells in one field. Okay, but with a high magnification, and that may not be required for your for addressing your biological questions. Okay, but really if you want to count, let's say, number of mitochondria per cell, okay, or you want to see the morphology of the mitochondria, okay, then you really need high magnification. Okay, so based on what you want to see or what you want to image, you have to decide that what magnification is required and what objective lens you will use. The next question is, what resolution do you need? Okay, it is possible that you may have a super resolution microscope facility in your lab or in your building. Okay, but that doesn't mean that for all your imaging experiments you will just use the super resolution microscope and not the regular fluorescence microscope. Okay, so how you can take that decision? So do you want to count the number of nuclear pore complexes per cell, or do you want to see their structures? Or do you really need super resolution imaging methods? That you have to address. Okay, so here you can see one figure I have given here. So the nuclear pore complex is labeled with some green color fluorescent dye. Okay, now the biological question that you are asking just need the counting of the number of nuclear pore complexes per nucleus, then I think this image is good enough for me. Okay, using the automated counting software, I can count that, okay, this nucleus has around maybe 200 or 300 nuclear pore complexes. Okay, I do not need to go into the super resolution to see their structure or morphology. I just need to count that how many these green dots are there, and each green dot represents one nuclear pore complex on the nuclear membrane. Okay, but let's say I really want to see the detailed structure of this nuclear pore complex. Okay, that how many foci are there in the nuclear pore complex. Okay, then I really need super resolution imaging to see the detailed structure of this nuclear pore complex. So actually, if you enlarge these small dots and you use the super resolution microscopy, then you can see only a smaller area of this cell. Okay, but you can see this kind of a resolved structures of the nuclear pore complex. Okay, so you have to decide the resolution, whether you really need high resolution for your imaging experiment or not. Then you have to ask, do you want to image live or fixed cells? Okay, because if you are imaging fixed cells, then phototoxicity or photo damage is not a concern for you. Okay, but if you are imaging a live cell, then phototoxicity is a concern for you. Okay, and you also have to check whether for your live cell imaging experiment, does your microscope has the environmental chamber to maintain the temperature, carbon dioxide, humidity. Okay, so those things you need to check, and you have to make the decision that whether you are going to image the live cell or the fixed cells. Then you have to check how fast do you need to image to capture your biological process. How fast do you need to image to capture a biological process? So how fast is your biological process that you want to image? Okay, let's say if I want to image a cell division, okay, and cell division generally takes 1 hour. Okay, so that is kind of a slow process. Okay, I can image at every 5 minutes for, let's say, 1 hour or even 2 hours to cover to capture the different stages of the cell cycle, like prophase, metaphase, anaphase, and how the cell is actually dividing. Okay, so just by taking one Z-stack image at every 5 minutes, I can actually capture that process of cell division. Okay, but let's say if I'm doing the single molecule imaging and tracking in live cells, right? And I have labeled the individual molecules of my protein of interest with a fluorescent dye. And those proteins move very rapidly inside the cell nucleus. Okay, so to capture those kind of fast movement, I cannot go for this kind of a time lapse that capture image at every 5 minutes. Okay, there I really need a very fast time resolution. Like I have to capture the images even at 15 milliseconds. Okay, so fast. Another thing you need to ask is how long do you want to image? So as I mentioned that if you want to study just the cell division, you just want to visualize one round of cell division, then I think 1 hour or 2 hour of imaging should be enough. Okay, because the cell division gets completed in 1 to 2 hours. Okay, but if you really want to see the embryogenesis, okay, entire process of embryogenesis, then you may have to image for a longer time, like in days. You have to image for even 1 day or even 2 days. Okay, so how long do you want to image? Because on that basis, you will also decide that which kind of fluorescent proteins you will need. Because your cells are live, you want to image them for let's say 1 day or 2 day. Okay, then you need those kind of fluorescent proteins which do not bleach so fast. Okay, because you will image them so frequently for such a long period of time. So you have to decide that how long do you want to image, whether you're, because see, the total duration of the movie can vary significantly from 15 seconds to 1 minute to 10 minutes to 1 hour to 12 hours to even days, several days. Okay, so that's why you need to figure out that how long do you want to image. The next question that you will ask is which fluorescent dyes or the proteins do you want to image? Are they photostable or do they blink? Why I mentioned do they blink? Because if you really want to go for the super resolution imaging, storm based super resolution imaging, you need a fluorescent dye that has the blinking property. Okay, so but if you want to go for other kind of super resolution or other kind of imaging, the blinking is not required. Okay, so you have to identify that what is your application and whether what are the fluorescent protein and the dye that you are using, whether that protein or the dye is compatible for what are the type of imaging technology that you are using. Okay, for storm imaging, you definitely need a dye which gives the photo blinking. Okay, then how many colors do you want to image? So in most of the applications, you can image 1 or 2 or 3 or maximum 4 colors together in the same sample. Okay, there are ways to image even more colors, but and spectrally separate them, but that is not the kind of a regular method that many people use. Okay, so mostly at max you can image 3 or 4 colors in the same sample. You by using let's say one dye in the ultraviolet region, the second dye at around in the green fluorescence, the third dye will give you red fluorescence, and the fourth dye will give you far red fluorescence. Okay. Then how many dimensions do you want to image? So the image can be captured in 2 dimensions, just X and Y, just one image is enough, or you need the Z information also, that is 3 dimension. Then you may need multicolor in Z stack, so that is a fourth dimension. Then you may need multi-time point or the time-lapse movie in multicolor, so that is a 5-dimensional image, and you may need multi-positions. Okay, so that is a sixth dimension. So you have to identify that in how many dimensions you need your image. Okay, then you also need to check what parameters do you want to quantify and how and using which software, because sometimes you have to take into account that how you can produce a better quality images which can be automatically segmented by your image processing software. Okay, so how efficient your image processing software in for the automated, let's say, segmentation or let's say automated tracking of single molecules. Okay, and how you can generate a better quality images so that it makes the life of the software easy to do its job. Okay, so those parameters you should also consider. Okay, then you have to decide which field of view is ideal for your experiment. Okay, so suppose you just want to see or want to count 100 cells and see their morphology and see whether they are different in colors or different in size and shape. Okay, so you need a larger field of view in which you can see so many cells. Okay, then only from one image you can easily count hundreds of cells and you can make that decision. But if you do over magnification for imaging this kind of equation, then you will see an enlarged image of the smaller area, the smaller field of view. Okay, so you can hardly see a few cells, and for counting 100 cells you may need to take at least 10 more images, okay, to count 100 cells. So here the field of view or the more magnification is not required for this kind of application. Okay, even with this magnification you can count the number of cells and you can get even more cells in one field. Okay, so this is what the field of view can be represented. If you want to see this entire group, this is a larger field of view. Okay, but the smaller field of view can show only a few faces. Okay, but here you can identify the faces, but in this you cannot identify the faces. So that's why you have to decide that what is more important for you, whether you just want to count the number of students in this group, okay, or you really want to see their faces. Okay, so if your biological question that you are asking, it requires that kind of a identification, then you should go for the higher magnification and take more such images so that you can get to see all the group members. Okay. Different companies manufacture this camera with different field of view. So here you can see some of the companies which kind of field of view they offer. It has been shown. Okay, so also while procuring the microscope, you should take care of this field of view before ordering the camera. Okay, because if you go for a camera with a very small field of view, and then for your some applications if you need a bigger field of view, it is not possible. So consider this parameter while procuring a new camera or a new microscope. The next major question is how to adjust the correct exposure time and intensity of the light source. Okay, so let's say you just put your sample on the microscope stage and you just focus your cells, and now you switch to the fluorescence mode. Okay, and then you will start seeing your fluorescence signal, something like this. Okay, but how you will ensure that, okay, whatever the exposure time or whatever the light intensity that you are using, that is correct and that is not too high or not too low. Okay, so for that all the softwares provides you this kind of a graphical representation of pixel intensity value. So you can see on the x-axis of this graph, it's a pixel intensity, and pixel intensities are from 0 to 1. Okay, so the 0 indicates that those are the dark pixels, right, the black pixels, and the right side, that is 1, indicates the bright pixels. So in this image, more the fluorescence intensity, this graph should go towards the right side. So let's say the same cell captured with a lower exposure time and higher exposure time. So if you go for very high exposure time, then you will see that your pixels are actually saturating. So many pixels are even accumulating at 1. That means those pixels are saturated. Okay, so ideally you should set the intensity of the light and the exposure time so that none of your pixel should be saturated, because from this image you cannot get the quantitative information, or you should not use this kind of image for the quantitative analysis, because some of the pixels which have already reached this saturation, they will underrepresent the actual fluorescence intensity. Okay, and this limit is imposed by the camera bit depth. Okay, so if the camera is 8 bit, then it provides 256 shades. Okay, so depending on the camera bit depth, this pixel intensities can be captured. So that means if you have a 16-bit camera, it can provide you the more dynamic range to collect more different more levels of fluorescence intensities. Okay, but if you are using an 8-bit camera, it will provide you a small dynamic range in which you have to adjust your fluorescence intensities. Okay, so ideally your this graph of pixel intensity should look like this. It should be mostly towards left, and some of the pixels may go up to 0.6, but some of the pixel intensity you should leave, because let's say you are adjusting your brightness and you are adjusting your exposure time and the light intensity based on this image. But let's say if you move to the next field, you may find some cells slightly brighter than this one. So if you have kept this thing, this area of the pixel intensity, then it's possible that for the next image, some of the pixel values may reach up to 0.8. Okay, so that's why I will just read through these statements so that you can understand it better in a better way. So avoid saturation and lack of dynamic range during setting up your exposure time and the intensity of the light source. So set the exposure time or the intensity of the light such that the resulting images use as much of the dynamic range of the camera as possible, but without saturating any images. Okay. Aiming for your image maximum to be 50 to 75% of the dynamic range is a safe bet. So what it is showing, this is a dynamic range, right? So from 0 to 1, that is your dynamic range. And you should set your exposure or the light intensity in such a way that 50 to 75% of the dynamic range is used. So from 0 to, let's say, 0.75. Okay, but you should keep some of the pixel intensity value available, because it might be possible that if you move to the next field, you may find some cells slightly brighter than this image. Okay, if you expect one sample in your experiment may be brighter than the other, a positive control, you may want to use it to determine your exposure setting to decrease the likelihood of ending up with saturated images. So what it is telling, let's say suppose you have two samples, let's say wild type and mutant, and you expect that the wild type should show you more fluorescence intensity compared to mutant. Okay, so in that case, while setting up the exposure time, you will put your wild type sample, which shows you the highest fluorescence intensity. Okay, so set the exposure time considering the wild type strain, which is expected to be more bright in fluorescence. Okay, and then you image your mutant, which is expected to be low in fluorescence. Okay, but don't do vice versa. Like you set this dynamic range or you set your exposure time and the light intensity based on the mutant. Okay, and then when you will image the wild type cells, and then you will start seeing the pixels are saturating, because the exposure time that you set for a mutant was actually high. Okay, and in the wild type, you will start seeing some saturated pixels like this. So that's why for adjusting this exposure time and the intensity of the light, you should first use the sample which shows you the highest fluorescence intensity. Okay, maybe a wild type or maybe a mutant. Now, how to choose an objective lens? So as we already briefly discussed in the first slide, that how you choose the objective lens, there are different types of objective lens available. Okay, 20x, 10x, 40x, 60x, 100x. Okay, but as I mentioned that it's not always the thing that if you have the 100x objective, so for all the applications we just go with 100x. Okay, because it provides the highest magnification. But on the other hand, you should also consider the numerical aperture of the objective. And why? So let's suppose your microscope is equipped with 200x objectives. Okay, there are two objectives of 100x magnification. One is with high numerical aperture, the other one is with low numerical aperture. So which one is better for your application? So you will say that obviously the objective with high numerical aperture, because it provides better resolution. Okay, but let's say for addressing your question, the resolution is not a concern actually. Okay, you really do not want to separate the two tiny spots which are very close to each other. Okay, what you want to say is you just want to count that how many nucleus or how many nuclei are present in this sample. Okay, so in that case, you should go for a objective lens with a low numerical aperture, because here you can see the high NA objective have a very small depth of field. If you have to image the entire nucleus, you have to take the Z-stack image with a high numerical aperture objective to collect the light from the entire nucleus. But if you go for the low numerical aperture objective, okay, then the depth of field will be high. So maybe the entire nucleus can be seen in a single focal plane. Okay, so you do not need to take the Z-stack. So let's say your objective is just to count the number of nuclei in the field. Okay, you just go for the low N objective where the resolution doesn't matter. You will definitely see the two nuclei separate because they are far apart. Okay, and you do not want to resolve them. So higher the high NA, high numerical aperture, that means the low depth of field, and the low numerical aperture that gives high depth of field. Okay, so the decision of using the objective lens based on numerical aperture. Okay, also how numerical aperture affects the brightness of your sample, because high numerical aperture objective lenses can collect more light from the sample. Okay, so if your sample is not bright enough, okay, let's say you are imaging a very faint sample, okay, in that case you need high NA objective so that it can collect the maximum light coming from the sample. But let's say you are imaging a sample which is very bright, then you can even image with low NA objective, because even some low NA objective will not be able to capture all the light coming from the sample, but it will at least collect a small portion of it, but that is also enough to make an image. Okay, so brightness of the sample also depends, or the selection of the objective also depends on the brightness of the sample. Whether your sample is bright, then go for low NA objective. If your sample is faint, then go for the high NA objective. Okay. Then this slide is specifically on when to use confocal microscope, because most of the time I have seen that people do not have a clear understanding about how confocal will produce a better image than the wide field imaging, and some misconceptions are generally there that okay, confocal will give better resolution. Okay, but that's not correct. So confocal should be used only for the thick samples, and thick means which are less than 100 micrometer in thickness. Okay, if they are even more thicker than 100 micrometer, then you should go for 2 photon illumination or light sheet imaging. But let's say if the samples which are less than 100 micrometer, the confocal is good. Okay, and conventional wide field microscopy collects both in focus and out of focus light, and to get rid of out of focus light, you can use the confocal microscope. So here I have shown these two images. This will be the difference between the wide field and the confocal image. Okay, and you can see that here the information is little hazy, because in wide field microscope it also collects the out of focus light along with the in focus light. Okay, so you can see your objects. Okay, here also you can see there are two blue whatever the nuclei. Here also you can see two nuclei. Here also some red color structure is there. There you can also see two red color structures are here. Okay, so if your objective is just to count the red and blue color objects in the cell, then even this image is enough. Okay, you can count the structures from this kind of image also. Okay, but if you want to see some more quantitative information, okay, or maybe you just want one good image for the representation purpose, the way here it is shown. Okay, let's say if I have to put one paper representative image in the paper in the publication or in my any presentation or report, I may just generate one good image using the confocal to show this kind of things, a better image. But let's say for my regular counting, this image is good enough. Okay, so confocal does not improve resolution over wide field. Okay, so thin samples have no benefit. So make sure that thin samples, generally the confocal will not give you a better image with the thin samples. Okay, and it cannot increase the resolution. And as I mentioned that for thick specimens which are greater than 100 micrometer, you should go for 2 photon or light sheet imaging. Now coming to another important point, that you should check the available microscope technologies and specifications in your facility. Okay? Because every time it is not possible that, let's say based on the questions that, so it is possible that let's say based on the questions that I showed you on first slide, you may come up with a very sophisticated imaging technology that you may need a 2 photon imaging for imaging your sample. Okay, but it is possible that that microscope is not available in your facility. Okay, so now how will you deal with it? Okay, so always there are some alternative options. If you do not have the best available technology in your facility, there are ways to use other kind of microscopes, at least to address your biological question. Okay, so what you should check in your imaging facility, which microscopes are available, whether you have wide field, confocal, turf, 2-photon, light sheet, which kind of microscopes are available in your facility. Then which light sources are available, LEDs, lasers, metal halide. Okay, then which filters are available, because you have to make sure that whatever the fluorescent dyes that you are using in your experiment, the appropriate fluorescent filters and LEDs or the light source should be available in your microscope. Because there are so many different varieties of filters tubes are available, right, for different fluorophores like DAPI, CFP, YFP, GFP, FITC, TRITC, M-cherry, Sapphire. So there are so many different colors and the filter tubes are available for different colors. So you have to ensure that which filter tubes are available in your microscope. Then you should check which objective lenses are available and their numerical aperture and immersion media. Let's say 100x objective is mostly available with the oil immersion, okay, but you may find the 100x with water immersion, or even 100x objective with glycerin immersion, okay, and 100x air objective you may also find. Same way for 63x, it is available in oil immersion also, as well as in water immersion, air immersion. Okay, so make sure that you check the immersion media of the objective lenses. Don't use any, let's say, water immersion or air objective with the immersion media, otherwise you will spoil that objective lens. Okay, so make sure the immersion medium of the objective lens, and whether that objective lens is a short working distance objective or the long working distance objective. Because if it is a long working distance objective, you should not put the objective too close to your sample, and eventually you will either break the objective or you will break your sample. Okay, then you have to check whether you have the live cell imaging chamber to maintain the temperature, CO2 and humidity. Because sometimes you may have a very good microscope, but if the live cell imaging chamber is not there, or even sometimes, let's say, the CO2 cylinder is not there, then you cannot maintain those environmental parameters. Okay, so ensure that you have the live cell imaging chamber to maintain these environmental parameters. Then which camera or detector do you have? Okay, whether SCMOS, EMCCD, CCD camera, or the photomultiplier tubes if you have a confocal. Then how fast can the microscope acquire an image? Okay, so if, let's say, you are really looking for fast imaging, you have to ensure that the microscope configuration is such that it can give you that speed. Okay, because camera itself takes some time to process the image, which is known as the camera processing time. So let's say you want to image at every 10 milliseconds, okay, but camera takes at least 20 milliseconds to process your image. So you cannot go for 10 millisecond time interval. Okay, you have to minimum give that camera processing time so that camera can capture one image and process it. Okay, so that takes, let's say, 20 seconds. So it is not possible to image lower than 20 millisecond with that system. Then do you have the automated X, Y and Z stage control? Because in some microscope you will see that they are not motorized. It's X, Y and Z stage is manually controlled. Okay, it is not motorized. Some microscope you will see that only the Z position is controlled, motorized. Okay, but not the X, Y. And some microscope you will find all X, Y and Z motorized. Also, you have to see the hardware and software features for image acquisition, autofocus and image processing. Suppose if you want to go for long-term time-lapse imaging, then you have to make sure that your microscope and the software has the appropriate options of for autofocus. Okay, and you have all the options or the features for image acquisition and image processing. Okay. So all these things you have to check that whether all these things are available in your facilities, and accordingly you have to design the experiments. And here are some general tips. So you don't always need a very expensive microscopy to answer a question. Sometimes using the simple microscopy, simple fluorescence microscopy, you can address most of the questions. Be aware of potential trade-offs between resolution, speed and photoimaging. So if, let's say, if you really want to achieve the high resolution, then you have to compromise on speed and you have to compromise on photoimaging. If you really want to go for very high speed imaging, then you may have to compromise on the resolution and photoimaging. Okay, so these things, that's a trade-off between achieving best resolution, highest speed and least photoimaging. You can fix a lot of problems with post-processing, but it's better to fix them during data collection. Okay, so see, these days there are several software modules available which can be used to make the image better after acquisition. Okay, so you may think that, okay, let me just capture the crap image and then do the deconvolution to make it look better. But that's not the correct way. Try adjust your imaging parameters or your microscope in such a way to collect the best quality data, and then you use whatever the image processing software you want to use, whatever like deconvolution or filtering, whatever you want to use it. But that doesn't mean that you even if you could have captured better quality images, but you didn't focus on capturing the better quality images because you are more relying on the post image processing. Okay. Never save your images in .jpg format. That is, loses the information. So never save your imaging data into .jpg format because you will lose some information. And the best way to store your images is in the manufacturer's proprietary formats. Like if you are using the Leica microscope, you should always keep the original proprietary format of your imaging data. Okay, whatever the microscope manufacturer you are using, like Leica, Zeiss, Olympus, Nikon, anything. So always keep a file in their proprietary formats like .lif, .nd2 for Nikon, .cgi for Zeiss, .olf for Olympus, and .lsm for Zeiss. Okay, and at least you should keep that data in .tiff format without any compression. Okay, so that the original information will remain. All the intensity values will be intact. If you want to do some quantitative analysis later on, then you can use the .tiff format for any kind of quantitative analysis in the future. That's it for today's class, and thank you so much for attending this course, and wish you all the best for your future fluorescence microscopy endeavors. And feel free to get back to me if you have any questions or concern even in future. Okay, thank you.